Welcome, everyone, and thank you for joining us for the first AGRO Lunch and Learn webinar of 2025. This webinar series is made possible through the generous support of Eurofin's AgroScience Services, and we sincerely appreciate their continued sponsorship. I'm Solito Sumulong, an executive committee member of the AGRO division and chair of the Lunch and Learn webinar committee. It's a role I've been honored to hold since 2021. Now, before introducing our speaker, I'd like to highlight the benefits of becoming an AGRO member. Membership enhances your science, expands your professional network, and supports your career growth. If you are not already a member, you can add AGRO to your existing American Chemical Society membership or join as an affiliate for just $20 a year, regardless of ACS membership status. Simply visit agrodiv.org and click Become a Member. Another important update, the ACS Fall National Meeting is coming up in Washington, D.C. this August on the 17th. The call for abstracts is now open and submissions are due by March 31st, so the end of the month. If you have programming questions, please reach out to our program chair, Dr. Ming Ma. The AGRO division is also committed to engaging new and existing members, enhancing communication and building future-ready resources for the global agrochemical community. Kevin, Edmund, Leah, and the AGRO team need your help. Please visit agrodiv.org to get involved. A quick note on today's webinar format. Please use the Q&A tool to submit your questions and comments, which we'll address at the end of the presentation. Also, let us know in the chat where you work and what you do. We'd love to hear from you. Gareth is very interested, so am I. So now it's my pleasure to introduce our speaker, Dr. Gareth Thomas. Gareth is a postdoctoral research scientist at Rothamsted Research in the UK, specializing in chemical ecology, particularly in agricultural and forest pests. His research focuses on the complex chemical interactions between plants, insects, and microorganisms to develop sustainable pest management strategies. He holds a PhD in biosciences from the University of Exeter and a bachelor's in biology from the University of York. I first met Gareth at the ACS Fall Meeting in Denver last year, where he gave a fantastic presentation in my mode of action session. We're all looking for new modes of action in the marketplace. So today he's going to kindly expand on that talk for a global audience. Please join me in welcoming Dr. Gareth Thomas to present the chemical ecology of acute oak decline. Gareth?
Thanks again for the very kind introduction there. It's a great pleasure to speak to you all today on the lunchtime seminar series, although in the UK it's slightly closer to dinner time. But yeah, it's great to be here, and I've been involved in the AGRO sort of committee for quite a while now in organising some of the symposium for the ACS annual fall meetings. It's been a great experience, great to be involved. And I'd encourage anybody who's interested in organising a symposium to definitely do so, because it's been a great experience. But yeah, I'll move on to the topic of my talk now, which is the chemical ecology of acute oak decline. So just a bit of an introduction to acute oak decline. So oak trees support a wealth of biodiversity and provide a habitat for a range of insect and animal species. But as with all plant species, they're subjected to a range of biotic and abiotic stresses, which can lead to the subsequent onset of disease. And one particular disease, which is particularly prevalent in the UK at the moment, is acute oak decline. And sort of the key symptoms of acute oak decline, if we look at this middle panel here, we can see we've got an oak tree here, which has got these kind of lesions which are coming down the trunk. And these lesions at the surface kind of ruin the aesthetic of the tree, but they can also go sort of deeper into the tissue and cause problems for the development of the tree. And it's quite a sort of, I'd say, it's an acute oak decline. It can lead to the death of the tree within sort of 3 to 5 years. So it can act pretty rapidly as well. So it's definitely an area of concern. And if we zoom in a bit closer here to one of the lesions, we can see that there's sort of a weeping fluid which is coming out of the lesion. And some previous work, which was done by our collaborators at Forest Research in the UK, has looked at the sort of microbiome, or the pathobiome, which causes this disease, and found there's 3 bacterial species, Brunneria goodwinii, Brunella victoriana, and Gibberella quercenicans, which are very commonly associated with these lesions, and are actually shown to be causing these lesions in the case of Brunneria goodwinii and Gibberella quercenicans. So, yeah, basically one of the key symptoms of these lesions, which are caused by bacteria. And in terms of the current distribution of acute oak decline in the UK, we can see that there's currently, it's fairly localised to the southeast of England, although we're starting to see a sort of northward shift in the disease onset, and it's been sort of detected regionally in Manchester and Liverpool, which is fairly north of the UK. So it's quite interesting that we're seeing this sort of northward spread. And one of the kind of key questions that is being asked is, why are we seeing this northward spread? So as well as the symptoms that are caused by the bacteria, there are also signs that beetles are very commonly associated with this disease. So you can see here on the first panel, this is what we call a D-shaped exit hole, which is very characteristic of acute oak decline. And this is caused by the beetle Agrillus bigotatus, called the jewel beetle because it's got a very sort of nice metallic colour. And in sort of over 90% of cases where we see the bacterial symptoms of acute oak decline, we also see the presence of this insect as well, which suggests there's quite a strong link between the beetle and the bacteria. If we look at this middle panel here, we can see where the red arrow is, we can see a bacterial lesion. And this blue arrow is sort of what's caused by the beetle larvae, and these are called larval galleries. And we've also got a picture here of 2 of the larvae outside of a tree. So yeah, as I say, there seems to be a very high co-occurrence between the beetles and the bacteria, which suggests there's potentially a link between the 2 in the disease onset. And the kind of research question that we're wanting to ask in this work is, what is the role for chemical signals in this beetle-bacteria interaction? So most of my talk today, I'm going to be talking about volatile organic compounds, and then later on in the talk, I'm going to be talking a little bit more about sort of chemical elicitors, which are produced by the larvae. So to provide a bit of context for volatile organic compounds and their role in ecological interactions, they're these low molecular weight compounds which can be emitted by plants, microbes, and animals. And these compounds can play a key role in communication. They can facilitate interactions between these organisms, for example, in host location, so plants can produce them, which allow insects to detect their food source, or potentially guiding oviposition as well. So insects can use these volatiles to know where to lay their eggs. And with volatiles, there's a definite potential for behavioral manipulation. So you can exploit these volatiles to influence insect behavior, and these can be applied sort of for monitoring insect pests as well. So you can use it for a monitoring tool or a pest management tool. So bringing it back to kind of the objectives of this work in terms of the chemical ecology of this syndrome, we're going to kind of divide the talk into 3 parts. So the first part is going to be focusing on leaf volatile organic compounds. We're going to assess if the tree disease status affects volatile production. So do healthy trees and diseased trees produce different volatiles from their leaves? And secondly, determine whether these differences in volatiles produced by disease versus healthy trees have any influence on the Agrillus bigotatus beetle behavior. Secondly, I'm going to be focusing on bacterial volatiles. So as I mentioned, these 3 causal bacteria of acute oak decline. We're going to determine the role for these bacterial volatiles on the Agrillus bigotatus beetle behavior, and characterize the volatile production from these bacterial species. So see what volatiles are being produced and are they having any role on beetles' behavior. And then finally, this is just going to take 1 slide, this last bit. We're going to talk a bit more about larval elicitor molecules. So whilst the majority of the talk is going to be the role of bacterial chemical signals on insect behavior, in the last part, we're actually going to look at the reverse and look at how insect chemical signals can influence bacterial growth and pathogenicity. So, first part of the talk, as I say, we're going to focus on the leaf volatile production. So this is the setup that was performed. So there's various sites around the UK where oak trees were identified as being healthy or symptomatic, across 6 different UK sites. And this is just an example of a dynamic headspace collection or an air entrainment kit where these volatile collections took place. So you can see here that quite simply, we use a cooking bag, which we can enclose around the foliage of the oak volatiles. At the bottom of this picture here, we've got a tube which contains a poropack, which is an adsorbent polymer. And over time, the volatiles are collected from this foliage. and can be analysed by gas chromatography and coupled gas chromatography mass spectrometry. Basically, we can sort of look at all this data as a whole from the healthy versus the declining trees and see, are there any differences in the volatiles produced by a healthy versus a declining tree? What was quite interesting here, as you can see by this graph, is that there seems to be 4 compounds which seem to be upregulated on a symptomatic oak tree, so ones that have got acute oak decline. So yeah, we've got 4 compounds here. We've got the mean compound production on the x-axis, and we can see that for all the symptomatic oak trees, all of these compounds are significantly upregulated by symptomatic oak. So that does indicate to us that symptomatic oak trees do indeed have unique volatile profiles compared to healthy trees. The next question we wanted to ask was, is there a role for leaf volatiles on beetle behaviour? So the first experiment we set up, this is a technique called forearm olfactometry, where we introduce different stimuli to the Agrillus bigitatis beetles. In this particular setup, we've got a blank air, which works as a control, and then simply just oak foliage on its own. So this is from a healthy oak tree. And we basically put the beetle into the center of this chamber here and record the amount of time it spends in each of these arms. So it's got 1 treatment arm, which is the oak foliage volatiles, and 3 blank control arms. And over several replicates, we can record the amount of time it spends in each treatment arm, and that can give us some indication as to whether there is a sort of attraction towards oak foliage volatiles. Again, this is just healthy oak. And what we've shown is, if we look at the mean time spent by virgin female beetles and virgin male beetles, we can see that there's a significantly greater amount of time spent by the beetles in those arms which contain the oak foliage volatiles compared to blank air. So that seems to be the case with virgin females and the virgin males. So we can conclude from that virgin female and male beetles show significant preference towards oak foliage volatiles. So these foliage volatiles are seen as a sort of host location cue. We slightly modified the setup for this next experiment though, and rather than looking at just healthy oak foliage volatiles on their own, we actually included these 4 compounds which are associated with acute oak decline into the headspace of one of the arms. So we basically got sort of healthy control treatment and then a treatment which contains oak foliage plus these sort of stress-related signals. And then we repeated the experiment for the forearm olfactometry. And when we introduced the Agrillus bigitatis virgin females into this setup, we can see that they've spent a significantly greater amount of time in the arm which contains the headspace from oak foliage plus the stress blend compared to a blank air control and an oak foliage control. So what that tells us is that the virgin female beetle shows significant preference towards the oak foliage volatiles plus this stress blend. And what this really tells us is that these virgin female beetles can potentially sniff out a diseased tree from the volatiles being produced, and that they would prefer to orient towards a diseased tree than a healthy tree, which is quite interesting. So that's all on the leaf volatile side of things. I'm going to focus on the bacterial volatiles. So we're looking at the actual bacteria which cause this particular disease. And as I mentioned in the introduction, we've got 3 species which seem to be associated with acute oak decline, which are Bruneria goodwinii, Rhinella victoriana, and Gibbsiella querceneckens. So we've got pure culture isolates for each of these individual strains, which were streaked out onto agar. We also included a control group, which is just growth media without any bacteria on it. And then we performed dynamic headspace collection. So a similar technique to the one we used to, well, it's the same technique as we used to capture the leaf volatiles, but just in a slightly different way. So we basically confined the bacteria in these glass chambers, and charcoal purified air was pushed over the headspace of these bacterial cultures. And then air was simultaneously drawn out of the top of the headspace through this poropaque powder, and all these bacterial volatiles basically get trapped onto this polymer within a glass tube. And then after 20 hours, we can basically stop the experiment, take the tube, pass a solvent through the tube, and this now allows us to elute volatiles into a liquid extract. And with this liquid extract, we can then perform a range of chemical and biological studies. And the thing we want to start off with is to determine whether these bacterial volatiles influence the behavior of these beetles. So this is a technique called forearm olfactometry. I appreciate some people will know what it is, but for those who don't, I'll give a kind of brief overview. It's basically a choice test which we use to determine insect preferences towards different stimuli. So the setup looks something like this. So hopefully you can see the cursor we've got this circular arena where we've introduced the beetle here, and then we've got 4 treatment arms. So 3 of the arms contain just solvent on its own, which is the control. And then one of these arms contains the volatiles that we just collected using dynamic headspace collections. And yeah, we do several replicates and basically record the amount of time that the beetle spends in each of these different arms. I've got a video here on the right-hand side showing the beetle making the choice. It's not the most interesting video, but at least it shows the setup in action, and we can see there that the beetle is moving towards the bacterial volatiles. So some of the data we obtained from that study. So first of all, I'll start talking about the control, which is just agar on its own. So we collected the volatiles from the agar without any bacteria on it, and showed that the beetle spent no significantly difference in the amount of time the beetle spent in the treatment compared to the control arm. But for all 3 of the AOD-associated bacteria, Brunneria goodwinii, Rhinella victoriana, and Gibbsiella querceneckens, we can see that these gravid female beetles spent a significantly greater amount of time in the treatment arm compared to the control arm, which was quite interesting. So from that, we can conclude that gravid female beetles show a significant preference towards AOD bacterial volatiles. So we've basically established that there's a biological role for these compounds in insect behavior. What we haven't done yet at this stage is look at what these compounds are, basically in the headspace. So that was the next thing we did. We took the mixture of compounds that the different bacteria species were producing and analysed them using gas chromatography flame ionisation detection. So this is represented traces across 4 replicates. On the y-axis, we've got the abundance of each of these compounds, and on the x-axis, we've got the retention time. And anywhere that we see a peak in the bacterial treatments suggests that there is a compound being produced. So a couple of things to note. We've got one compound which seems to be abundantly produced across all 3 bacterial species. But what's quite interesting is that we've got these other compounds which are specific to the Brunneria goodwinii. And one thing to note about Brunneria goodwinii is that it seems to be the most pathogenic out of the 3 species, so the most in terms of disease-causing properties. So yeah, we've got 3 compounds of interest, compound 1 and then compounds 2 and 3, which seem to be being produced by the bacteria. And compounds 2 and 3 specific to Brunneria goodwinii, which was particularly interesting to us. And yeah, from that, we can conclude that there was one compound abundantly produced by all of the AOD bacteria, and then 2 compounds which were specific to Brunneria goodwinii. So that's all very interesting, but how do we know that the beetle is actually interested in these compounds? Well, we've got a technique that we use in the laboratory called gas chromatography electron sonography, which is quite a difficult word to say, but hopefully I'll explain the principle of it to you sufficiently. So we basically adopt the same principle as before. So we use gas chromatography to separate out the compounds in this mixture. But as the compounds are separated, the beetle's antennae, which you can see in this image here, gets exposed to the compounds as they're separated individually. And if the antenna can detect any of these compounds, it causes an electrical stimulus to occur. So you can basically link compounds in a complex blend to the antennal response. Hopefully when we see these traces, it'll become a bit clearer. So we've got 2 traces here for 2 of the bacteria, Brunneria goodwinii and Gibbsiella querceneckens. So what we can see, if you can follow my cursor on the bottom trace here, is this looks similar to the GC trace we saw beforehand. So we've got a compound 1 there, compounds 2 and 3, which are specific to the Brunneria goodwinii. Along the top is our EAG response. So we can see that there's some sort of peaks which are sort of coming off as the compounds come off. And in the case of compounds 1, 2, and 3, we can see that the beetle's antenna can detect these compounds. We also did this experiment for the Gibbsiella querceneckens bacteria, which was only producing compound 1. And again, we can see that the antenna does seem to respond to compound 1. So from this experiment, we can conclude that the volatiles produced by Gibbsiella querceneckens and Brunneria goodwinii elicit the antennal response in female Agrilla spigitatus. But one of the sort of key things to highlight about this technique, GCEAG, is that just because an insect's antenna is responding to a compound, it doesn't mean to say that the insect is necessarily going to be attracted to it. So it could be a sort of response that means that the insect would be repelled by it. So the next thing we need to do is go back to our old friend forearm olfactometry. But this time, rather than using the entire headspace that were collected by the bacteria, we actually managed to purify out the compounds using preparative HPLC and test these compounds individually in the olfactometer. So we can actually look at specific compounds and see, is the beetle interested in these compounds? So we'll start off with compound one. Compound one was the compound that's produced by all three of the acute oak decline bacteria. And we can see that the beetle spent a significantly greater amount of time in the treatment arm compared to the control arm. So that means that the beetle is indeed demonstrates a preference for compound one. We also purified out compounds 2 and 3. And again, when we put them in the olfactometer, we can see that the beetle spent a significantly greater amount of time in the arm containing these compounds compared to the solvent control arms. And then finally, to test something that was more representative of what the bacteria are producing, we took the 3 individually purified compounds and put them back in the olfactometer and found that the beetle preferred the arm which contained all 3 of the compounds compared to compound 1 alone and compared to the control. So that indicates that the gravid female beetle shows significant preference towards these purified EAG-active bacterial volatiles. And the reason, I should say, why we tested gravid female beetles is because in the life cycle of the beetles, once they've mated in the tree canopy, the gravid female beetles then move down the tree toward the bark to lay their eggs. So these gravid female beetles, which are egg carrying, would be the ones that would be more likely to detect these bacterial volatiles. I probably should have said that a bit earlier on, but yeah. So that's it for the volatile chemistry for the leaf and bacterial volatiles. For the final part of the talk, we're going to focus on the larval elicitor molecules. So we're going to examine the role of larval elicitors on bacterial virulence. So yeah, we're kind of flipping it on its head a bit now and looking at the chemistry from the insects and seeing, does that have a role in the bacteria's kind of pathogenicity. I think a lot of chemical ecology studies look at bacterial volatiles and insect behavior. But this is quite an interesting reverse of that to see, is there anything that we can learn from the insect's chemistry in terms of how the bacteria cause diseases. So to provide a bit of background to this study, what we did was we performed these oak log inoculation tests in the labs. These were interesting kind of semi-field assays where we inoculated logs with eggs of the Agrillus bigitatus larvae, and then kind of let them develop over time into larvae, and then collected the larvae from the tree and basically wanted to see what compounds were being produced. So we had 2 hypotheses. The first hypothesis was that the diseased wood can stimulate bacterial growth. And the second hypothesis, which is the one I'll focus on, is that the larvae produce chemical elicitors which can stimulate the growth of the bacteria. I should say here as well that we're focusing in this experiment just on the Brunneria goodwinii as the most disease-causing bacteria. So we're looking at the interaction between these larval chemical elicitors and the Brunneria goodwinii's growth. So for the third part, just an overview of the experiment, we've got a water control, and then we've got our different treatments where we've basically applied these chemicals extracted from the larvae into a bacterial culture and then monitored the growth of the bacteria over time, and also looked at gene expression changes in genes which cause disease. So I'll break it down a little bit because there's quite a lot of graphs on this slide. But one of the kind of key interesting things we found was that we can see that the growth of the bacteria, the Brunneria goodwinii, seems to be greater in the treatments which have got these larval extracts compared to the controls. And the growth rate is higher. I don't think there's a significant difference at this stage in the growth rate, but we can see that there's a trend towards it. And then when we actually count the number of bacterial cells in these treatments, at 0 hours post-inoculation, we see no significant difference, which we'd expect. But after 24 hours, we can see that there's significantly more bacterial cells in the treatments which contain this larval chemical extract compared to the control. So that's kind of indicates that the larvae do indeed produce chemical elicitors which can stimulate the growth of the Brunneria goodwinii bacteria. And the next thing we wanted to do is focus on the genetics behind this. So we focused on specific genes which the bacteria have, which cause pathogenicity. We focused on these three virulence genes here, two of which are involved in biofilm formation, and basically wanted to see, is there any difference in the RNA-seq transcript count per million across the treatment? So to give a bit more context, we wanted to see if is this a specific interaction between the Agaricus bigitatus and the Brunneria goodwinii. So we tested a range of chemical extracts collected from different larvae, different species, including Agaricus planopennis, which is the emerald ash borer, Agaricus salsicolus, and Agaricus bigitatus. And the key thing that we found was that if you look at the RNA-seq transcript count per million for these three virulence genes, we can see that there's significantly greater expression of these genes in the Agaricus bigitatus extracts compared to the other ones. And the reason why this is particularly interesting is because it does indicate that there's some specificity in this interaction between the beetle and the Brunneria goodwinii bacteria. So even when we've got these closely related species, such as Agaricus planopennis and Agaricus salsicolus, which are within the same genus as Agaricus bigitatus, they don't seem to elicit this increased putative virulence gene expression. And the fact that these genes are kind of upregulated by the Agaricus bigitatus extracts could indicate that the bacteria becomes more pathogenic in the presence of the larval extracts. This would need verifying under sort of open field conditions, but it certainly looks like it's sort of tending towards that way. So from that, yeah, we can conclude that the Agaricus bigitatus larval chemical elicitators stimulate the growth and pathogenicity of Brunneria goodwinii, and that this interaction does seem to be specific to Agaricus bigitatus. And just for people's interests, this work has been submitted for publication. We've also put it onto the preprint server bioarchive. If anyone wants to have a read of that in more detail, I've provided a link there, and I can provide a link in the chat as well after the talk. So, conclusions of this work. First of all, the Agaricus bigitatus can differentiate between healthy and declining foliage volatiles, and they seem to orient toward declining volatiles. So, yeah, the beetles can differentiate between a healthy and a diseased tree. The AOD bacteria produce volatiles, including two volatiles which seem to be specific to Brunneria goodwinii. Gravid female beetles show a specific preference towards these AOD bacterial volatiles, and these volatiles also elicit antennal responses in the beetles. And finally, elicitors from Agaricus bigitatus larvae can stimulate the growth and pathogenicity of AOD bacteria. So we provided some quite novel insights into the interaction between the beetle and the bacteria. Probably one of the most important slides is why are we doing this work? So there's a lot of sort of future work and applications which can sort of come of this. One of the things we'd like to do in the future is to assess the effectiveness of these bacterial volatiles at attracting Agaricus bigitatus under field conditions. So using one of these semiochemical based lures, which have basically got these attractive volatiles in the lures, basically see can we see that there's an increase in the amount of Agaricus bigitatus captured by this lure compared to the controls which don't have the lure. From that, we've also got an image here of somebody putting one of these lures into an oak tree. One of the benefits of doing this and having a sort of a blend of compounds which does seem to attract the Agaricus bigitatus is that we can possibly utilize these lures as tools to monitor the presence of the beetle populations effectively. So kind of use it as a proxy for the spread of the insect and potentially the disease further north across the United Kingdom and elsewhere. And finally, use it as a potential early warning system. So we can basically have these traps which are supplemented with these attractive volatiles, and then the forestry managers can kind of be alerted to the presence of the beetle, which could indicate that the acute oak decline is either present in their forest or is about to be present in their forest, and they can take management action to prevent this from happening. So that's it from me. I've got a lot of people to thank in the slide, so I usually spend a good time on this bit of the slide, particularly people at Rothamsted Research, particularly Joseph Vutz, John Caulfield, Mike Burkett, David Withall, Godfrey Apangu, and Pradip Songra have been instrumental in this work. Collaborators at Forest Research, particularly Sandra Denman, and collaborators at the University of Birmingham and the University of West of England. I'd also like to acknowledge the support that we received, the financial support from the UK Research and Innovation Strategy Priorities Fund, the Bacterial Plant Diseases Grant, which was funded by the BBSRC, the Department for Environment, Food and Rural Affairs, the National Environment Research Council, and the Scottish Government. And finally, to say that Rothamsted Research receives strategic funding from the Biotechnology and Biological Sciences Research Council, with knowledge funding from growing health and smart crop protection, which is also funded by the BBSRC. I'd also like to say a special thanks to Dr. Nuria Tabanka, who is very important in AGRO's work. And she's helped me to get involved in the AGRO division, and she's been a great co-organizer of lots of symposia over the years. So special thanks to her. Just to say as well, for anyone who uses LinkedIn, I've got, hopefully this QR code works. If you scan it, it should bring up my LinkedIn profile. So I'm happy to carry on the conversations further, either via LinkedIn or via my email address here. So yeah, that's all for me, and thank you very much for listening.
Thank you so much, Gareth. That was fantastic. It really expanded what we talked about here in Denver, or over in Denver last year. So thank you so much. We have an intelligent audience here, as evidenced by a lot of the questions that have come up. So to bundle a couple of questions that came in early, Gareth, can you tell us a little bit about the nature of these microbes, where they live, tendency to multiply and produce these VOCs?
Yeah, so what's recently been shown about these bacteria, which kind of emphasizes how important the last bit of the talk was, is that these are environmental bacteria. So they are present, and they're even present on trees which don't have acute oak decline, where we don't see the symptoms of acute oak decline. So some recent work has shown that, yeah, the three causal bacteria seem to be present even on healthy trees. So what we think is basically happening is that the kind of interaction between the beetle larvae and these bacteria basically switches the bacteria from being a kind of non-pathogenic on the tree to sort of a pathogenic form. So, under normal conditions, they don't seem to cause disease, but when the larvae is present, that's when it triggers them. It switches on these pathogenicity genes and possibly increases their growth. So we think that the beetle is kind of required for that to happen.
Right. I'm going to bunch a few remarks on the nature of the volatiles now. So we've had questions about the toxicity of the volatiles, whether volatiles change when larvae are present with pines infection changes, as well, the identity of the 3 oak VOCs as well. Could you and as well, as well as some remarks about that. So can you expand a little bit from those dimensions on those volatile organic compounds?
Yeah, so the volatiles, we're basically quite close to publishing this work, so I'm not able to say what the compounds are, but yeah, we kind of were able to purify them out. I don't know if they're toxic particularly to sort of in general. We just I think that they're just used by the beetles as a sort of way of locating their hosts. Yeah. And what was the other part of the question? Sorry, there was.
Yeah, I guess the other part of the VOC question, Gareth, would be how you select the absorbent materials for trapping the volatiles from the beetles as well.
Yeah, that's a really good point. Yeah, so there's a range of trapping methods we could use for trapping the volatiles. Poropack is the one that we use most commonly in the lab because it allows us to generate samples which can be kind of reused for behavioral and chemical tests. There are sort of more destructive methods like Tenax, which we could have used. Tenax has got the added benefit that we don't need to use a solvent, so we won't see, there won't be any compounds masked by a solvent peak, but it makes it a little bit more tricky for when we're doing behavioral tests. But yeah, so that's why we sort of opted for Poropack. I think that's SPEME as well. would have been a good one to use, but again, harder to work with for biological tests. There's one thing oh, sorry.
Do you use the polymer directly to analyze the volatile compounds?
So we use the polymer to sort of capture the volatiles through these dynamic headspace collections, and then once we've eluted the solvent, we're basically left with this solvent extract, which we then sort of use for those tests.
Right.
I should also say, I don't think I mentioned this in my talk, but we've collected lesions from Acute Oak Decline trees, just basically chopped the lesion off a tree, and we're actually able to see the production of the compounds from the lesion when it's actually on a tree. So I know that what we've shown is the compounds being produced by growth media, which is not a very realistic environment. The bacteria are going to be sort of grown in much sort of nutrient poorer conditions compared to this nutrient-rich media, but we have successfully found that these compounds are also produced from the lesions themselves. So yeah, that's another thing to add there.
I'm going to try to do you a favor. Can you back up to the reference, the slide with the one reference, so I can dump that into the chat as well? Take a couple of seconds to do that. Yeah, I could do that. Oh, apologies if I botch the link.
Is it easy if I stop sharing my screen, or?
Here we go. It's on the chat.
Oh, great. That's great. Thanks very much.
Great. Onto another question on natural predation for these beetles. Would those predators also be would they also be attracted by those VOCs as well?
Yeah, that's something that I think some work has been undertaken on sort of identifying natural enemies for these beetles. Yeah, it's kind of hard to identify if there's a parasitoid wasp. There seems to be quite a high co-occurrence of these beetles with some parasitoid wasps, which could indicate that the wasps could be using these cues also to identify where the beetles are. But yeah, I think that works. It's not being done here, and it's sort of quite early days. So I don't know too much about that, but it's a really good point.
Yeah. Alan Felsot had a question about what advantages the beetles have in their preference. Have you examined carbon-nitrogen ratios to see if diseased trees may have lowered this ratio and therefore benefiting the beetles by a higher nitrogen dietary source? And I'll add a little bit as well on that. I know you are mapping out the northward trend of the prevalence of these diseases. Perhaps there could be an underlying trend from this ratio as well.
Yeah, no, that's a really good point. I think so. Yeah, this project I was part of, this bacterial plant diseases project. There's lots of different people working on different aspects of the disease, and there is a group who are looking into if there's like a nutritional benefit to the beetles, i.e., does the onset of the disease sort of change almost like the nutritional status of the tree and the carbon nitrogen ratio. I'm not sure what the results of that work is just yet, but they are looking into that. There's also the possibility that the lesions themselves, because the beetles seem to want to move towards them, they could have a higher sort of sugar content or higher amino acid content, which was beyond the scope of the work that we did, but I think is going to be taken forward in future work. So that's a really good point. Yeah.
Right. Yeah. A corollary to that question would be, is there anything you want to add as well on the link between the bacteria and the beetle? Any benefits between the two parties in the interaction, or any progress identifying the larval elicitor?
Yeah, so that's I'll start with the benefit to the beetle. That's kind of sort of follows on from the previous question. Is something that needs to be looked into further. Is there a nutritional benefit to the beetle for selecting these diseased trees? And yeah, interesting. What what's the larvae getting out of it as well. Yeah, to the point about the identification of the compounds, that's something that's going to take a lot of work. So we've not included that in this publication, but I think plans going forward is to focus on which compounds are causing this. I didn't speak about it because I didn't have time, but we've actually fractionated out the larval extract. This is in the publication as well, so if anyone wants to read that, they can. We've fractionated out the larval extract into 4 fractions and found that one of the fractions in particular is active. So we kind of narrowed down which compound or compounds could be causing this activity. And yeah, the next stage is to identify what the compounds are, which I'm told will take quite a lot of work. But definitely something that we're keen to look at because it's a very important research gap at the moment. So
yeah. Any new angles on the analytical approach as you move forward on the research? To go back to the question about isolating and characterizing the VOCs.
Yeah, so the VOCs, we've got our identifications for them. Yeah, as I say, we're hopefully going to be publishing the preprint in the coming weeks. So I can maybe speak to you to circulate that. Yeah, the larval list is though they're slightly more complicated molecules. They're sort of heavier molecules. So with this paper, we haven't sort of honed in too much on the identity of those compounds. We've got the data for it, and I think in follow-up work, that's where that'll come into it because yeah, it'd be really important to look at and really interesting.
Right. This is a great question from Christopher Leake, asking about the actual mechanism that kills the oak tree. Do you have any remarks about the mechanism or on the disease?
Yeah, so what I think basically happens is you get this sort of the lesions which form on the surface can actually go further into the tree, and I think they can disrupt the kind of water and nutrient flow. And you've also got loads of insects who are kind of forming these larval galleries. So it's kind of interrupting the sort of internal physiology of the tree, which then has impacts on its on its general status. I think another symptom which I didn't quite speak about in detail is that you also see sort of dieback of the tree crown as well. So you can kind of see the exposure of branches as they lose their leaves when the trees kind of get this disease. Well, one area which would be particularly interesting would be to see if we can, as we've shown a difference in the volatile profile between a healthy and a symptomatic tree, would be to kind of look at volatile changes as the disease progresses. So you'd almost need to set up an artificial experiment where you've got a healthy tree and a diseased tree, and you could track over time the volatile profiles of the leaf foliage as a sort of earlier indicator of disease presence, because I guess by the time you can see the lesions on the tree, it's already a little bit too late. So yeah, looking at a volatile detection system would be something that'd be really interesting.
Right. And I did write down a couple of questions on that, just since we do have plenty of time for questions. And please, if you have any other questions for Gareth, please put those in the Q&A tool. What would a practical solution look like based on the research? Is it about blocking VOCs? Is it about mimicking and luring with VOCs? Where do you see the future demand? Where do you see the future demand for your research? I know we talked about early warning systems. What's existing now in the market for curing this disease, and how do you see the research being a part of the ecosystem here?
Yeah, no, that's a really good question. So yeah, we've had some market research done on this sort of system, and it doesn't look like there's anything in place at the moment to in terms of monitoring tools for this specific pest. I know other monitoring tools do exist based on pheromones and things like that, which might not necessarily always be effective. So yeah, in terms of the practical application of this work, there's two ways. Well, there's a few ways of looking at it. I think the first one, as I mentioned, would be kind of a monitoring tool. So including these bacterial volatiles into these semi-chemical based lures to improve the monitoring of the beetle. But one of the key things to highlight is that this is a native beetle, so we're not interested in getting rid of it necessarily. But there could be instances where we've got a particularly high prevalence of these beetles and the disease, where it might be an idea to kind of selectively sort of place quite a lot of these semi-chemical based lures around. So we're sort of honing in on one area where we're capturing more of the beetles, rather than sort of doing a kind of blanket thing like that all over the United Kingdom. So yeah, I think as improving the monitoring tools would be good. As I say, for this current system, I don't think any exists. But maybe a good example would be definitely over in the USA and Canada. There's a high prevalence of the emerald ash borer, Agrilis planopennis, and I think they've got some commercial laws over there for that. So following in that kind of footsteps would be quite interesting, and hopefully that's successful.
Now, from your perspective in the UK, it's great that we have you and your research highlighted within the AGRO division. How do you see any practical solutions for the agricultural marketplace?
Yeah, no, that's a interesting point. I suppose this is a specific example of how we can use microbial volatiles for forest pest management. But a lot of the work I'm doing at the moment is looking at the use of microbial volatiles for sort of agricultural pest management. So looking at agricultural pests like aphids, sort of things like that, which affect sort of food crops as opposed to agroforestry crops. Yeah, sort of using that as a platform to manage pests as well. So yeah, we're looking at above ground pests. I'd say aphids, quite a big one. There's a lot of work at Rothamsted that's being done on below ground pests as well. So wireworms are a pretty big problem in the UK and pretty much everywhere, as I understand it. And yeah, just quite interested in looking at how microbial volatiles can be used for pest management. As I say, this particular example is focusing on agroforestry, but also for agricultural food systems as well. I think there's quite a lot of potential there.
Yeah. And this is this last question for me. It's you're a biologist presenting with the AGRO division of the American Chemical Society, right? And we're so happy to be here with you today and this evening, really. And you are a biologist based in the UK for the American Chemical Society. Could you tell us a little bit about your relationship with the AGRO division and how you see your relationship growing within this community? This is a reason why I mentioned a few. I solicited our audience to tell us where you worked and what you do, so you get a sense for that as well.
Yeah. So yeah, as I said in my acknowledgements, I've been involved with AGRO sort of first of all as a presenter. So I was at the 2019 San Diego meeting presenting my PhD results, which Nuriat kindly invited me to. And then more recently, I've been involved with the actual organisation of the symposium with Nuriat. So she's been great at kind of mentoring me through that process. And I'm involved in the sort of communications and early career committees as well. So yeah, I've really enjoyed my time in the AGRO division. I think it's been good as well to try and increase the participation from other people within the institute. So more people from Rothamsted have come over to the AGRO division as a result, and they've all really enjoyed it as well. So yeah, no, I'd definitely encourage people to get involved, and it's a great community. And it's good because it kind of spans across a lot of different sectors. We've got academia, industry, policy, and I think over the years I've definitely learned a lot from everybody in the different kind of sectors. So yeah, it's been great.
Oh, fantastic. Thank you so much, Gareth.
Thanks
And
very
thank
much.
you to the audience as well. You guys have been fantastic. We have a diehard membership. We have a diehard audience here at the webinar series. So I thank you all very much for a very interactive session this morning and this afternoon here in the US. A special thank you to you, Gareth, for your excellent presentation, to Eurofin's Agroscience Services for their generous sponsorship, and to Kyle Sawyer, our dedicated webinar coordinator. For any questions that we didn't get to, we'll be posting responses in the coming days, so stay tuned for those updates. I'll also dump a link to the reference on the the YouTube web recording description as well. So be on the lookout for that. You may also receive a follow-up survey, and we greatly appreciate your feedback to help us continue improving this series. So mark your calendars. Our next webinar is on April 16th, featuring Megan Tietz from Smithers, who will present Exploring the Essential Ecotoxicology Tests for Environmental Risk Assessment. It's one of those big big elephants in the room when we do talk about our science. I think this will be a great way to explore some of the basic tests and get a decent context for the day-to-day of research that our division conducts on a daily basis. We also have additional webinars lined up on Wednesdays at noon in May, September, October, and November. The one on May 7th will be on USA data compensation, which is always another elephant in the room. And with our global audience, it's always good to get that perspective on this little quirk of registering pest control products in the United States. If you have any ideas for future webinar context, we'd love to hear from you. Please reach out to me. I'm the chair of the webinar committee and been that way since 2021. The AGRO webinar series is a fantastic way to introduce new audiences to AGRO's incredible community and showcase the groundbreaking science within our division. It's also a great opportunity for anyone interested in learning about new areas and even stepping in as a future moderator. We do have a guest moderator on May 7th who's certainly in it for the learning. So I'm really excited to introduce you to him as well. So thank you again for your participation, and we will see you on April 16th. Have a great month upcoming.